August 2024. Now at I am streaming my live study with me. It will start very soon. तो स्टार्ट जितना देर तक हो सके उतना देर तक ही करूँगा, ठीक है। Ribosomes are associated with the plasma membrane or they are about 15 nanometer. They are about in prokaryotic ribosome in Kaleki Yolo, as usual. That is the mesosome, special the mesosome which is from vesicle tubule lamella, vesicle tubule lamella, vesicle tubule, vesicles tubule, vesicles tubule lamella, vesicles tubules and lamellae, vesicle tubules and lamellae, vesicle tubules and lamellae, vesicle tubules and lamellae. They help in cell wall formation, replication and distribution of blood cells. They also help in respiration secretion process to increase the surface area of the plasma to increase the surface area of the plasma membrane to increase the surface area of the plasma membrane and enzymatic content. In some prokaryotes like cyanobacteria, there are other membrane restrictions in some prokaryotes. In some prokaryotes like cyanobacteria, there are other membranous extension into the cytoplasm called chromatophores, which contain pigments. Bacterial cell may be motile or non-motile. If motile, they have thin filamentous extension. Bacterial cell may be motile or non-motile. Bacterial cell may be motile or non-motile. If motile, they have thin filamentous extension. They have thin filamentous extensions from their cell wall called flagella. Bacteria show a range. Bacteria show a range in the number and arrangement of flagella. Bacteria show a range in the number and arrangement of flagella. Bacterial flagellum is composed of three parts. Bacterial flagellum is composed of three parts. Filament, hook and basal body. Filament, hook and basal body. The filament is the longest portion and extension from the cell surface to the outside. Besides flagella, pili and fimbriae are also surface structure of the bacteria, but do not play a role in motility. The pili are elongated tubular. The pili are elongated tubular structure made of a special protein. The fimbriae are small bristle-like fibers sprouted. The family are small vessel-like fibers sprouting out of the cell. Fibers sprouting out of the cell. In some bacteria, they are known to help attach the bacteria. They are known to help attach the bacteria to rocks in stream, and also the host tissue. Ribosome and inclusion bodies. In prokaryotes, ribosomes are associated with the plasma membrane. In prokaryotes, ribosomes are associated with the plasma membrane. In prokaryotes, ribosomes are associated with the plasma membrane. Ribosome in prokaryotes, ribosomes are associated with the plasma membrane of the cell. Plasma membrane of the cell. They are about 50 nanometer by 20 nanometers. The prokaryote ribosome are associated with the plasma. In prokaryote ribosome are associated with the plasma membrane. Ribosome are associated with the plasma membrane of the cell. Ribosome are associated. Ribosome are associated in prokaryote. Ribosome are associated with the prokaryote with the plasma associated with the plasma membrane of prokaryote cell. Ribosome ribosomes are associated with the plasma. Ribosome are associated with the plasma membrane. Ribosome are associated with the plasma membrane. With the plasma membrane. Ribosome are associated with the plasma membrane of the cell. Plasma membrane of the plasma membrane of the cell. Ribosome are associated. They are about 15 nanometer. They are about ribosome 15 nanometer by 20 nanometer. In size are made two subunits, 50s and 30s. Two subunit, 50s and 30s. Two subunit, two subunit. Units which may present together from 70s prokaryotic ribosome. Ribosome on the ribosomes are the site of protein synthesis. 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 Several ribosomes may attach to a single mRNA. Several ribosomes may attach to a single mRNA. Several ribosomes may attach to a single mRNA and form a chain called polyribosomes or polysomes. The ribosome of a polysome translates the mRNA into proteins. The ribosomes of a polysome. The ribosomes of a polysome translate the mRNA into proteins. The ribosome of a polysome translate the mRNA into proteins. The ribosome of a polysome translate the mRNA into proteins. The ribosomes of a polysome translate the mRNA into proteins. The ribosomes of a polysome translate the mRNA into protein. Inclusion bodies. These are naturally into Inclusion module get reserve material in prokaryotic cell. Reserve material in prokaryotic cell. Reserve material in prokaryotic cells are stored in reserve material in prokaryote. Reserve material in prokaryote. Reserve material in prokaryote. Prokaryotic cell. Prokaryotic reserve material in prokaryotic cell are stored in cytoplasm in the form of reserve material in prokaryotic inclusion module reserve material in prokaryotic cell. Reserve material in prokaryotic cell. Reserve material in prokaryotic cells are stored in the cytoplasm. Reserve material in prokaryotic cells are stored in the reserve material. Reserve material in prokaryotic cells. Plant, animal, and fungi. Plant, animal, and fungi. Plant, animal, and body plant is included in the eukaryotic includes all protein plants, animals, and fungi. In eukaryotic cells, there are in eukaryotic there is and in eukaryotic cell there is in eukaryotic cell includes all proteins. In eukaryotic includes all the proteins, plants, animals, and fungi. In eukaryotic cells. In eukaryotic cells, the eukaryotic body plant, animals, and fungi. In eukaryotic cells, there is an extensive compartmentalization of cytoplasm through the presence of membrane-bound organelles. Through the presence of membrane-bound organelles. Eukaryotic cells possess an organized nucleus with a nuclear envelope. Cells have a variety of complex locomotory. In addition, eukaryotic cells have a variety of complex locomotory and cytoskeletal structure. Their genetic material is organized into chromosomes. Their genetic material is organized into chromosomes. Their genetic material is organized into chromosomes. Eukaryotic cells have a variety of complex locomotory. In addition, eukaryotic cells have a variety of complex locomotory and cytoskeletal structure. Their genetic material is organized into chromosomes. All eukaryotic cells are not identical. Plant and animal cells are different. Plant and animal cells are different as the former possess cell wall as the former. Plant and animal cells are different as the former possess cell wall. Former possess cell wall, plastids, and a large central vacuole which are absent in animal cells. On the other hand, animal cells have centrioles which are absent in almost all plant cells. For the cell wall plastic and a large central vacuole which are absent in animals. Animals in absent key, animals in absent central vacuole which are absent in animals. Cell wall plastic, large central vacuole. Cell wall plastic and large central vacuole, gas vacuole central, which are absent animal cell. Structure. The detailed structure of the membrane was studied only after the detailed structure in 1958 spectral microscope. Studies on the cell membrane, especially in the human red blood cell, enabled the scientists to deduce the possible structure of plasma membrane. This study showed that the cell membrane is mainly composed of lipid and proteins. The major lipid are phospholipid that are arranged in a bilayer. Phospholipid that are arranged in a bilayer. Also, the lipids are arranged within the membrane. The lipids are arranged within the membrane. Within the lipids are arranged. The lipids are arranged within the membrane. The lipids are arranged within the membrane with the polar head towards the outer side and the hydrophobic tail. Hygroscopic. জল ধরে রাখা হচ্ছে জল গিয়েছে ওর নাম হচ্ছে কি আবার স্ফ্যাগনাম পিটমস. Also, the lipids are arranged within the membrane. Also, the lipids are the biochemical investigation clearly revealed that membranes also possess protein and carbohydrate. The ratio of protein and lipid varies considerably. The ratio of protein the ratio of protein and lipid varies. The ratio of protein and lipid The ratio of protein and lipid varies considerably. Protein and carbohydrates. The ratio of protein and lipid. The ratio of protein. The ratio of protein and lipid varies considerably in different types. The ratio of protein and lipid varies considerably in different types in inventing the membrane of the erythrocyte. The membrane of the erythrocyte has approximately 52% protein and 40% lipid. Depending on the case of extraction membrane, protein can be classified and integral. The peripheral protein lie on the surface of membrane, while the integral proteins are partially or totally buried in the membrane. An improved model of the structure of cell membrane was proposed by a Singer and Nicholson. It is widely accepted as fluid mosaic model. According to this, the quasi-fluid nature of lipid enables later movement of proteins within the overall bilayer. This ability to move within the membrane is measured as its fluidity. This ability to move within the membrane is measured as its fluidity. The fluid nature of the membrane. The fluid nature of the membrane is also important from the point of view of function like cell growth, formation of intercellular junctions, secretion, endocytosis, cell division, etc. The fluid nature of the membrane is also important. The fluid nature of the membrane is also important from the fluid nature of the membrane. The fluid nature of the membrane is also important. The fluid nature of the membrane. The fluid nature of the membrane. The fluid nature of the membrane. The fluid nature of the fluid nature of the membrane. The fluid nature of the membrane is also important from the point of view of function like cell growth, formation, intercellular junction, secretion, endocytosis, cell division. One of the most important function of the plasma membrane is the transport of molecule across it. One of the most important function of the plasma transport of the molecules across it. The membrane is selectively permeable. The membrane is selectively to some molecule present on the other side of it. Many molecules can move briefly across the membrane without any requirement of energy, and this is called passive transport. Neutral solutes may move across the membrane by the process. Solutes may move across the membrane. Neutral solutes may move across the membrane by the process. Neutral solutes may move across the membrane by the process of simple diffusion. Neutral solutes may move across the membrane by the process of simple diffusion along the concentration. that is from the from higher concentration to the lower. Okay. The polar molecule cannot pass through the non-polar by lipid bilayer. They require a carrier protein. They require a carrier protein of the membrane to facilitate their transport across the membrane. A few ions or molecules are transported. A few ions or molecules are transported across the membrane. Again, they require a carrier protein of the membrane. They require a carrier protein. They require a carrier protein of the membrane to facilitate that. They require a carrier protein. They require a carrier protein. They require a carrier protein of the membrane to facilitate their transport across the membrane. A few ions or molecules are transported across the membrane against their concentration gradient. That is, from lower to a few ions or molecules are transported across the membrane against their concentration gradient. That is, from lower to the higher concentration. Such a transport is an energy dependent process. Such a transport is an energy dependent process in which ATP is utilized and is called active transport, e.g. in a plant's potassium pump. Oh, sorry, why? Sorry. As a transport, active transport, sodium, potassium. Okay. As the polar molecule cannot pass through the non-polar lipid bilayer, as the polar. Yes. As the polar molecule cannot pass through, as the polar molecule cannot pass through, as the polar molecule cannot pass through the non-polar lipid bilayer, they require a carrier protein. Membrane. They require a carrier protein of membrane to facilitate their transport across the membrane. A few ions or molecules they require a carrier protein of the membrane to carrier protein. A few ions or molecules are transported across the membrane against their concentration, that is from lower to higher concentration. Such a transport energy dependent, yes, ATP utilized and active transport, sodium potassium pump. Okay, cell wall. Cell wall which, as you may recall a non-living rigid structure called the cell wall from and cell wall forms an outer covering for the plasma membrane. Plasma membrane of fungi and plants. while each of the membrane some of its structure and function, many of these are considered electron microscopy studies electron microscopy studies of eukaryotic cell electron microscopy studies of eukaryotic cells reveal electron microscopy studies of eukaryotic cell reveal the presence of network. Electron microscopy studies of eukaryotic cell reveal the presence of a network or reticulum of tiny tubular structure scattered in the cytoplasm that is called endoplasmic reticulum. Electron microscopy studies eukaryotic cell reveal the presence of a network or reticulum of tiny tubular structure is scattered in the cytoplasm that is called the endoplasmic reticulum hence endoplasmic reticulum divides the intracellular endoplasmic reticulum. The presence of a network or reticulum of tiny tubular structure scattered. Presence of a network or reticulum of tiny tubular structure scattered in the cytoplasm that is called. Hence, endoplasmic reticulum divides intracellular space into two distinct compartments, luminal inside extraluminal and extraluminal cytoplasm compartment. The endoplasmic reticulum often show ribosome attached to their outer surface. Endoplasmic reticulum shows ribosomes attached to their outer surface. The endoplasmic reticulum, the endoplasmic reticulum bearing ribosome, the endoplasmic reticulum bearing ribosomes on their surface. The endoplasmic reticulum bearing ribosome on their surface is called rough endoplasmic reticulum area. The endoplasmic reticulum, the endoplasmic reticulum often show ribosome attached to their outer surface. The endoplasmic reticulum bearing ribosome on their surface is called the endoplasmic reticulum bearing ribosome. Their surface is rough endoplasmic reticulum. In the absence of ribosome, they appear smooth, called smooth endoplasmic reticulum. Area is frequently observed in the cell. Area reticulum is frequently observed in the cells actively involved in protein synthesis and secretion. They are extensive and continuous with the outer membrane of the nucleus. They are extensive They are extensive and continuous. They are extensive and continuous with the outer membrane of the nucleus. The smooth endoplasmic reticulum is the major site for the smooth endoplasmic reticulum as major site for synthesis of lipid in animal cell. Lipid steroid hormone synthesis in animal cell. Lipid steroidal hormones are synthesis in ACR. The smooth endoplasmic reticulum is the major site for synthesis of lipid. The smooth endoplasmic ACR. ACR. ACR is the SAR small molecule is the major site for synthesis of lipid. In animals, major site lipid synthesis of lipid major site synthesis of lipid. Synthesis in lipid in animal cell lipid steroidal hormones are synthesis initial in animal cell lipid steroidal hormone is synthesis initial. हेलो भाई जो भी देख रहे हो थैंक यू. Golgi apparatus. Camille Golgi 1898 first observed densely stained reticular structure densely. Camille Golgi 1895 observed densely stained reticular structure near the nucleus. Reticular structure near the nucleus. Reticular structure. Camille Golgi first observed densely stained reticular structure near the nucleus. These were later named Golgi bodies after him. They consist of many flat disc-shaped sacs or cisternae of Golgi 1898 first observed densely stained reticular structure near the nucleus. These were later named Golgi bodies after him. They consist of many flat disc-shaped sacs or cisternae of 0.5 micrometer to 1.0 micrometer diameter. These are stacked parallel to each other. Varied number of cisternae are present in a Golgi complex. The Golgi cisternae are concentrically. The Golgi cisternae are concentrically The Golgi cistern are concentrically arranged. The Golgi tale, these are stacked parallel to each other. Varied number of cisterni are present in a Golgi complex. The Golgi cisterni are concentrically arranged near the nucleus. Concentrically arranged near the nucleus with distinct convex cis or the forming face and concave trans or the maturing phase, the cis and the trans phases. Cis and concave trans are the maturing phases. The cis and the trans phases of organelles are entirely different but interconnected. The interconnected. The cis or trans 77. Okay. Phase and concave trans or the maturing phases. The cis and the trans phases of the organelle are entirely different but interconnected. The Golgi apparatus principally perform the function of packaging. The Golgi apparatus principally perform the function of packaging materials. The vacuole is the membrane bound space. Vacuole is the membrane is found in cytoplasm. It contains water, sap, excreted product, and other material not useful for the cell. Vacuole is bound by a single membrane called tonoplast. In plant cells, the vacuoles can occupy 90% of the volume. In plants, tonoplasts facilitate the transport of number of ions and other material against the concentration gradient. The vacuoles, hence their concentration is significantly high. Engulfing the food particles, engulfing the mitochondria. Mitochondria হচ্ছে গিয়ে find mitochondrion singular হচ্ছে গিয়ে mitochondrion useless unless specifically stained are not easily visible under the microscope. The number of mitochondria per cell is variable depending. The number of mitochondria per cell is variable depending on the physiological activity of the cell. In terms of shape and size also. Considerable degree of variability is observed. Typically, it is sausage shaped. Considerable degree of variability is observed. Typically, sausage shaped. Hey, what's up? 14. Only subsequent states are not easily visible under the microscope. The number of mitochondria per cell is variable depending on the physiological activity of the cell in terms of shape and size. Also, in terms of shape and size, also considerable degree of variability is observed. Typically, it is sausage-shaped or cylindrical, having diameter 0.2201 micrometer. Average 0.5 meter and length 1 to mitochondrion to 4.1 micrometer. Each mitochondrion is a double membrane mitochondrion is a double membrane. Mitochondrion is double membrane structure with the outer membrane with the outer membrane and the inner membrane dividing it with the outer membrane and the inner membrane dividing it lumen distinctly into two aqueous compartments. That is the outer compartment, and in the inner compartment. The inner compartment is filled with the dense homogeneous substance called the matrix. The outer membrane forms the continuous limiting boundary of the organelle. The inner membrane forms a number of infolding. The inner membrane forms a number of infoldings called cristae, singular crista toward the matrix. The cristae increase the surface area. The two membranes have their own specific enzymes associated with the mitochondrial function. The two membranes have their own specific the mitochondrial function. Mitochondria are the site of aerobic respiration. Mitochondria are the site of aerobic respiration. They produce cellular energy in the form of ATP. Hence, they are called powerhouse. ATP mitochondria is the powerhouse of the cell. Mitochondria is the powerhouse of the cell. The matrix is also possess single circular DNA molecule, a few RNA molecule, ribosome 70, and the component required for the synthesis of the components required for the synthesis of protein. The mitochondria divide by fission. The mitochondria divide by fission. Plastids are found in all plant cells. Plastids are found in all plant cells. In euglenoids, these are easily observed under the microscope as they are large. They bear some specific pigment. Thus imparting specific colors. Imparting specific colors to the plant. Based on the type of pigment, plastic can be classified into chloroplast, chromoplast, nucleus, nucleus. The chloroplast contain chlorophyll. The chloroplast contain chlorophyll and carotenoid pigments which are responsible for the chloroplast contain chlorophyll. The chloroplast chromoplast nucleus. Based on the type of pigment, plastic can be classified into chloroplast, chromoplast, nucleus. The chloroplast contain chlorophyll and carotenoid pigments which are responsible for trapping light energy, essential for photosynthesis. In the chromoplast, fat should be carotenoid. Fat should be carotenoid. In the chromoplast, fat soluble carotenoid. Fat soluble carotenoid. Okay. In the chloroplast, the chloroplast contains chlorophyll and carotenoid pigments which are responsible for trapping light energy essential for photosynthesis. In the chromoplast, fat soluble carotenoid pigment like carotene in chromoplast. In chromoplast, the chromoplast fat soluble carotenoid pigment. Fat soluble carotenoid pigment like carotene, xanthophyll, and other are present. This gives the part of the plant a yellow or red color. The leucoplasts are the colorless plastids of varied shape and size with stored nutrients. Amyloplasts stored carbohydrates. Starch. Example: potato. Content. Amyloplast. Amyloplast stored carbohydrates. Starch. Amyloplast stored carbohydrates. Amyloplast stored carbohydrates. Amyloplast stored carbohydrates. Starch. Amyloplast stored carbohydrates. Starch. Example: potato. Amyloplast stored carbohydrates. Amyloplast stored carbohydrates. Starch. Example: potato. Amyloplast stored carbohydrates. Example: elioplast. Elioplast store oils and fats, whereas amyloplast stored carbohydrates. Starch. Example: potato. Amyloplast stored carbohydrates. Starch. Potato. Or elioplast stored oil and elioplast stored oil and fats, whereas the aleuroplast amyloplast, alioplast, amyloplast, alioplast, aleuroplast, amyloplast, amyloplast, amyloplast stored carbohydrate, starch, potato, alioplast stored oil and fat, alioplast stores oil and fat, alioplast stores, alioplast store oil and fats, whereas the aleuroplast store protein, store proteins, whereas the aleuroplast store proteins. Majority of the aleuroplast store protein. Majority of the chloroplast of the green plants are majority of the chloroplast of green majority of the chloroplast majority of the chloroplast Majority of the chloroplast of the green plants are found in the mesophyll cell. Aleuroplast store protein, aleuroplast store protein, aleuroplast store protein, aleuroplast store protein, amyloplast store carbohydrates, alioplast store oil and fats. Alioplast stores oil and fats. Alioplast stores oil and fats. The chloroplast which contain chlorophyll, carotenoid pigments. The chloroplast which contain carotenoid. Chromoplast contain carotenoid pigment like keratin, xanthophyll, and other present that give the color. Part of the plant yellow, orange, red color. The leucoplast which is colorless, plastid and light shape and size. Store nutrients. Store leucoplast which is nutrients. Leucoplast which is store leucoplast store nutrients. Leucoplast which is nutrients. Leucoplast store nutrients. Amyloplast, and leucoplast store leucoplast which is store nutrients. amyloplast, alloplast আর হচ্ছে কি তোমার amyloplast কি করে amyloplast হচ্ছে কি তোমার কার্বোহাইড্রেট স্টোর কার্বোহাইড্রেট স্টার্চ পটেটো আর alloplast হচ্ছে কি স্টোর হচ্ছে oil and fat আর aleuroplast হচ্ছে প্রোটিন majority of the majority of the majority of the hey bro, what's up? हनी डिट्स कैसे हो भाई? कल भी तुम बात किए थे मेरे साथ. Majority of the chloroplast of the green plant. Majority of the chloroplast of green plant. Majority. भाई आप पढ़ो पढ़ो पढ़ो. अगर कुछ आपको आई एम ऑल्सो गुड. आपको कुछ पूछना है तो मेरे कम्युनिटी पोस्ट पे जो भी पोस्ट है उस पर कमेंट कीजिएगा, आपसे बात कर लूँगा. आप पढ़ाई के टाइम स्टडी स्लॉट इसमें अगर बात नहीं करोगे तो अच्छा ही होगा. थोड़ा बहुत चलेगा, लेकिन आपको स्पेशली कुछ पूछना है तो पूछो. बहुत ज़्यादा तो नहीं आ रहा है, लेकिन आ रहा है ठीक ठाक. आपको ठीक है थैंक यू. पढ़ाई करो अच्छे से. आई विश आप अगले साल मेडिकल कॉलेज में ही दिखोगे. थैंक यू. Majority of the chloroplast of green, majority और बहुत बढ़िया नंबर आ रहा है. ऐसे ही करते रहो. आपको तो अगले साल पक्का एम्स मिलेगा. अब ऐसे ही करते रहो. Majority of the chloroplast of green plant, majority. ओके भाई, ठीक है. आप पढ़ाई करो. शायद आपको अब ज़्यादा डिस्ट्रैक्ट मत हो, इधर उधर मत करो. ऐसे ही पढ़ते रहो. आपको बहुत अच्छा मार्क्स आ रहा है. ओके ब्रो, ठीक है. आप पढ़ो. ठीक है, लाइव एंड कर रहा हूँ. आपको